Hi students, welcome back to another session. Today we will be discussing the topic nucleic acid from the chapter Biomolecules. So last class we discussed about the nucleotides, the biomicromolecule. Today we will discuss about biomacromolecule nucleic acids. Let's start. So what are nucleic acids? Nucleic acids are called the molecular blueprint of life. Can anybody say why it is called the molecular blueprint of life? Because nucleic acid that consists of chemical substances or chemical composition that transmit that consist of these chemicals are the genetic information that transmit from one generation to another generation. That's why this nucleic acid is called a molecular blueprint of life. It is essential for the life. Then where can we see nucleic acids? Where can we see nucleic acids? Nucleic acids are present generally in the nucleus of the cell. Okay, so nucleic acids are very, very essential to the life and it is called a molecular blueprint. It is present in the nucleus of the cell. The major function, nucleic acid control all cellular functions. Inside the cell, various chemical reactions, various activities, metabolic reactions taking place, which is under the control of nucleic acid. And also it controls the heredity, transmission of genetic information from one generation to another. That's what heredity. Okay. And then it helps in the synthesis of protein. Protein synthesis, the data and all carried by the DNA, and DNA will give the message to the to the RNA, and from there only protein synthesis taking place. Okay, correct. Yes. So I hope this part is clear. What is nucleic acid, where it is found, and what are the vital functions of nucleic acid. Now, what are the different types of nucleic acids? Can anybody say what are the different types of nucleic acids? What are the different types of nucleic acids? Yes. DNA and RNA are the two types of, correct. DNA and RNA are the two types of nucleic acids. So, DNA is what? DNA is deoxyribonucleic acid, and RNA is what? RNA is the ribonucleic acid. Let's compare these two nucleic acids, DNA and RNA. DNA, it is the primary genetic material in most of the organisms. Most of the organisms, all the genetic material is present in the DNA. The genes are present in the DNA, which is transmitted from one generation to another. But in the case of RNA, RNA is the genetic material of some viruses. We know the viruses are classified into what are they? DNA viruses and RNA viruses there. RNA viruses. Okay. So, for example, COVID-19, that virus is coronavirus is an example of RNA virus. Okay. So, that such kind of viruses, the genetic material is the RNA. Now, when you compare the structure of the DNA and RNA, DNA is a double stranded structure. That means there is one strand and also another strand, like this double stranded structure only DNA. Coming to RNA, RNA is only single stranded structure. It's a single stranded polynucleotide. And in the DNA, name itself, you can see the difference. It is deoxyribonucleic acid. What is that? Because it consists of the sugar is deoxyribosugar. In the previous class, I've told you what is the difference between the deoxyribosugar and the ribosugar, right? So deoxyribosugar, in which carbon atom the oxygen is removed? In which carbon atom? The second carbon atom oxygen is removed. Okay. So DNA consists of deoxyribosugar and RNA consists of ribosugar. And in the DNA, there is a permanent information storage. Okay, but RNA is active for the synthesis of protein. So, when you compare DNA and RNA, which is more stable? DNA is more stable than RNA. And what are the reasons? This is important question. The reasons for more stability in DNA than RNA. One reason is presence of deoxyribose sugar, because there is one functional group is less only in this DNA. If there is less functional group, it is more stable. And then the nitrogen base also some difference is there, right? What are the nitrogen bases present in the DNA? Yes, adenine, thymine, guanine, and cytosine. But in RNA, adenine, uracil, guanine, and cytosine. So when you compare uracil and thymine, thymine is more stable. That is another reason the DNA's stability. So is that clear, the differences between DNA and RNA? Right? So now we will discuss in more detail about the structure of the DNA. Okay, so in the previous class, this is just a recap only. In the previous class, we learned the building block of DNA nucleic acid, that is nucleotides. We know that the components of nucleotides, they are phosphate group, pento sugar, and a nitrogen base. This is the structure. So basically, this is that furanous form of the sugar. And in the first carbon atom, what is attached? The nitrogen base is attached, and the fifth carbon atom, phosphate group, is attached. Okay, so there are many nucleotides joined to form the polynucleotide chain, that is the nucleic acid. Okay, so here second nucleotide, here the phosphate group attached, then third one, in the first carbon atom, nitrogen base is there. In the fifth group, fifth carbon atom, phosphate group is attached. Like this, there is a long chain of nucleotides that form the polynucleotide chain. Why I am telling about polynucleotide chain here? Because nucleic acids are made up of polynucleotide chain. Okay, so here see, in the suppose this is a first nucleotide, this is second, third, like that the last one. Okay, so in the first nucleotide, the third carbon atom, first, second, third carbon atom, OH group is attached with the phosphate group, and here you can see two ester bond, so it is called a phosphoester bond. Okay, so remember the two the nucleotide chains are joined with the help of phosphodiester bond. Similarly, here also, the third carbon atom OH group is attached to the phosphate group. And then next, okay, like this it will go. So first, there will the first carbon atom, first carbon atom, nitrogen base is attached. Okay, and the third carbon atom to the second one, the phosphate group. Like that, the polynucleotide chain will be formed in the case of nucleic acids. Remember the difference between the nucleotides and nucleic acids and nucleosides, right? Yeah. So let's start with the double helix model of the DNA. Okay, the structure of the DNA. So can anybody say what are the different types of DNA? There are different types of DNA present. Okay, A DNA is there, B DNA is there, C DNA, D DNA, like that different DNA is there, and these DNA and all double helical structure only, but its helical structure is right-handed form. Right-handed helical structure, this A, B, C, D. ZDNA is the left-handed form. Okay, left-handed form, left-handed helical structure only, ZDNA. And here we are discussing about the double helix model that was explained by two scientists, Watson and Crick, in the year 1953. Remember, in 1953, Watson and Crick explained the DNA model that is known as the double helix model. And out of this 5 different types. Watson and Crick explained about the bDNA. Okay, so remember, this is also one important question. Watson and Crick explained what type of DNA, that is the bDNA. Okay, so now what is double helix and what is exactly there inside the bDNA? That's what we are going to discuss. So the DNA consists of 2 polynucleotide chain. Okay, so there is one polynucleotide chain is there, and that is the coiled separately, and there is another polynucleotide chain coiled in such a way that it looks like a spiral staircase. Okay, so according to Watson and Crick, bDNA consists of 2 polynucleotide chains, and they are arranged in such a way that it looks like a spiral staircase. Okay, and when you check the chains, 2 polynucleotide strands, they are arranged anti-parallelly. Very important point. Okay, anti-parallel arrangement. What do you mean by anti-parallel arrangement? The opposite direction only the 2 polynucleotide chains are arranged. Okay, that means in the previous slide we discussed how to make the polynucleotide chain, right? So there is the first nucleotide here, and this is free only. Okay, so if we'll go back to the diagram here, so here you can see the first nucleotide in the 5th carbon atom, it is free. It is not attached with any of the other group because this is the very first one. Okay, and when you check the last nucleotide of the polynucleotide chain, the 3rd carbon atom is free. So this strand we can call as the 5' 3' chain. Okay, 5' 3' chain. See, you have to learn this topic very clearly because for next year, both biology and in chemistry, this is important. Okay, the structure of a polynucleotide chain and the structure of the DNA. So, nucleotide, see, one nucleotide is arranged like this. Okay, like this it is arranged. So, suppose this is the first and this is the last one. The 3rd carbon atom will be attached with the 5th carbon atom of the subsequent nucleotide. Here also it is attached to that 5th one. Okay, so when you compare like this, the first nucleotide, the 5th carbon atom is free. So we can say this is the 5' end. And in the last one, first, second, the 3rd one supposed to attach to the next phosphate group of the next nucleotide. That is not there. So here the 3', this end is called a 3' end. Okay, so because the 3rd carbon atom is free. Then the opposite strand, one more strand is there. How the strand is arranged, it is in the opposite way, like this. Okay, and this is the first carbon atom, 2nd, 3rd, 4th, then the 5th one. Okay, so here nitrogen base is attached. Okay, then here another one like this. Here nitrogen base, 2nd carbon atom, 3rd, 4th, then the 5th carbon atom. Okay, like that it is arranged. First carbon atom, 2nd, 3rd, 4th, 5th. Okay, now first, 2nd, 3rd one, 3rd carbon atom, here it is free only. Okay, and in the 5th carbon atom, here the phosphate group is attached with the first nucleotide and the subsequent nucleotide. Similarly, here the 3rd carbon atom is attached to the previous one. Okay, like this, the opposite way the 2 nucleotide chains are arranged. So here you can see the 3rd carbon atom is free only. So this is called a 3' end. And coming to the last nucleotide, the 5th carbon atom, 1, 2, 3, 4, 5. Okay, the 5th carbon atom is free. It is not attached with any of the phosphate group of the subsequent nucleotide. So this is the 5' end. Okay, that's what the 2 strands run Anti-parallelly, one will be in the 5' to 3' end, the other will be 3' to 5' end. Are you getting my point? Yes? Yeah. So, DNA looks like a spiral staircase. Okay, remember a spiral staircase, like this, a spiral staircase. So, in the staircase, we know there are steps, right? Steps are there and a railing is also there. The step represent here, they are the nitrogen base pairs, right? Nitrogen base pairs. And railing is actually formed of sugar phosphate. Sugar phosphate backbone that forms the railing of the DNA staircase. Okay, so the nitrogen base form the steps of the adjacent. So this is one nucleotide nitrogen base will be attached with the another nucleotide's nitrogen base. Okay, so sugar phosphate will be the backbone, like this in the staircase. Sugar phosphate like that, the that form the backbone, and the steps here, it is made up of nitrogen base such as adenine, thymine, then guanine, cytosine, like that. The steps are there. Okay, like this. So here we know the previous class. Do you remember adenine pairs with the thymine? How many bonds? There are 2 bonds, right? Then what bond? What is the name of the bond? The name of the bond is hydrogen bond. Okay. Yeah. So remember all those features, key structural features of DNA: anti-parallel arrangement, sugar phosphate backbone, nitrogen base form bases, that pairing form in the form of steps. Then there are complementary base pairing. This one we will discuss again in the next slide. And some measurement you have to remember. Okay, so come back to this diagram. See, there is one complete turn. Suppose if a person is standing here in the spiral staircase, first complete turn till here. Okay, so till here, this complete turn, this is called one pitch. Okay, and the distance in one pitch, that is called a 3 point. The length of a length per turn or length per pitch is called 3.4 nanometers or 34 angstrom. Remember these numbers. Okay, so 34 angstrom or 3.4 nanometers. Sometime in the option they will give, instead of nanometer, angstrom only. Okay, then so what is pitch? The complete one full turn in the spiral staircase, that is called a one pitch. Okay, and the length of one pitch is 3.4 nanometer. Now you check how many numbers of this base pairs are there in one pitch. Okay, 1, 2, 3, 4, 5, 6. Here 1, 6, 7, 8, 9, 10 base pairs are there. Okay, so one pitch consists of, there are 10 base pairs. We are discussing about which DNA? We are discussing about the bDNA. Okay, so in the bDNA, one pitch consists of 10 base pairs. Correct. Yes, what you said, it is correct. 10 base pairs are there. Okay, so we know the length, the complete one pitch, it is 34 angstrom. Suppose in angstrom it is 34 and it consists of 10 base pairs. Tell me what is the distance between the adjacent base pairs. You know the everything, the information I have given you. 10 base pairs, 34 angstrom. So the distance between the adjacent base pair is, yes, the distance between the adjacent base pair will be, it is, yeah, 3.4 angstrom. Okay, 3.4 angstrom or 0.34 nanometer. Remember that also. So the distance between the adjacent base pair is equal to 0.34 nanometer or 3.4 angstrom. Clear? Then the diameter also you need to remember. The diameter of the DNA, that is 2 nanometer. Okay. And how many bonds are there in the DNA? Can anybody say what are the different types of bonds present in the DNA? Yesterday, last class, we learned about the number of bonds present in the nucleotides. Okay, so here we have to add some more bonds name. Which are the bonds present in the DNA? Yes, you are right. What are the different types? Glycosidic bond is there, right? N glycosidic bond is there. Then what else? Phosphodiester bond is there, and in between the nitrogen bases there are hydrogen bonds also there. Okay, remember hydrogen bonds there, phosphodiester bonds there, N glycosidic bond is there. Okay, so I hope there is no doubt in this part. Shall we go to next slide? Yes. Okay. So last class we discussed this topic, Chargaff's rule. Again, one more time. Chargaff's rule, base pairing. Chargaff explained a base pairing rule, that is in 1950. According to Chargaff, there are certain points you have to remember. Chargaff's rule. According to Chargaff, adenine always pairs with the thymine and vice versa with the help of two hydrogen bonds. Okay, then guanine pairs with the cytosine and vice versa with the help of three hydrogen bonds. So here the strong is more stable, stronger bond, right? Stronger bond is here. Another point, according to Chargaff, the amount of purines and pyrimidines are equal. Purines you remember, right? Adenine and guanine. Adenine plus guanine, that is equal to the pyrimidines, thymine and cytosine. Then adenine is equal to thymine. Guanine is equal to cytosine. Okay, so base pair ratio from one species to another species will be different. But the base pairing, that ratio will be same in all the individuals in this specific species. Okay, so remember all those points. Chargaff's rule, Irwin Chargaff explained about the base pairing, and this pairing will be acceptable or applicable only in the case of DNA double stranded one, not in the RNA. So this also another important point. Chargaff's rule explaining only the DNA structure, not in the RNA, because we are discussing about the base pairing, right? RNA is a single strand. There is no need of base pairing. Okay, so this complementary base pairing ensures the amount of purine always equal to the pyrimidines in the double stranded, which maintain a uniform diameter of the double helix. So assertion, reason, question, chances there. The last statement. DNA maintain a uniform diameter of the double helix. How? Because there are complementary base pairing. One purine always pairs with a equal that pyrimidines. Okay, so the number of purine and number of pyrimidines equal only. Because of that, the DNA can maintain a uniform diameter throughout the chain. Related to this, you can expect some questions. Okay, so according to the Chargaff's rule, there is one question. Please, you have to do this question right now. In humans, almost 30 percentages adenine present. Adenine is 30 percentage. What is the percentage of other bases? Other bases. I'll give you 2 minutes. You have to find out the percentage of other bases. So the question once again, in humans DNA, there are 30 percentage adenine present. Find out the percentage of other bases. Yes, fast you have to do. How can we find out the percentage of other nitrogen base? Yes, Eva said it is 20 percentage. How did you get 20 percentage? Any other answer? Okay, so we know that adenine is equal to thymine, right? Number of adenine and a number of thymine is equal. Adenine, if it is 30 percentage, then thymine also will be 30 percentage, right? So adenine, thymine 30 percentage. That means out of 100 already 60 over, right? Then guanine and cytosine 40 percentage. Okay, altogether 40 percentage. That means guanine is equal to cytosine 40. Out of that, in that 20 will be guanine and 20 will be cytosine. Okay, I hope Eva explained. It is like mentioned that is a guanine and cytosine, right? 20 percentage. Okay, 20 percentage. So total, and that adenine, thymine, guanine, cytosine, like this way. Okay, so you can expect this way questions. Okay, so that is related to Chargaff's rule. Coming to the next is RNAs. We know that RNA is a single stranded structure and it is mainly involved in the protein synthesis. Okay, RNA involved in the protein synthesis specifically and also the gene regulation. So here we just discuss about the different types of RNA. Not much explanation about the RNA. That next year you will study more about the types of RNAs, specific RNA functions and all in the molecular biology part. So types of RNA, there are three different types of RNA mainly: messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA or rRNA. Messenger RNA, messenger. Okay, so messenger RNA out of the total cellular RNA, when you compare, only 5% present. And this messenger RNA carries certain message in the form of, that is the genetic code. Okay, genetic code or the message from the DNA will be given to the ribosomes for what purpose? For the production of protein synthesis. That message will be given from the DNA to the ribosomes to the messenger RNA. Okay, so protein synthesis, these these different types of RNA play important role. Messenger RNA giving messages to the ribosomes from the DNA. Then the transfer RNA, okay, transfer RNA, they are transferring or they are bringing specific amino acids to the ribosomes during protein synthesis. We know the proteins consist of amino acids. So the different units of amino acids are distributed in the cytoplasm. So those cytoplasm should be taken and will be given to the ribosomes by the transfer RNA. Why all are giving to ribosomes? Because ribosome is known as the protein factory of the cell. Okay, so it is a site where the protein synthesis is taking place. So ribosomes require the message and the raw materials. Okay, and also for speed up the reaction, there are enzymes required that will be that will be given by the ribosomal RNA. That means the ribosomal RNA acts like a ribozyme enzyme. Okay, that help for the peptide bond formation. What are peptide bonds? The bond formed between the amino acids. Okay, amino acids, many amino acid join to form what? Many amino acid join to form the polypeptide. Okay, that is a protein formation. So the bond formed between the amino acid is a peptide bond. So the peptide bond formation enzymes required. That enzyme as the ribosomal RNA. Is it clear about the types of RNA? Yes. So types of RNA we discussed. Then the structured double helix model of the DNA, differentiation between the DNA. Okay, there are certain extra information that is not there in the NCERT textbook, but we should know about some extra other RNA types. Okay, so some other RNA type, one is called the snRNA, small nuclear RNA. That RNA is involved in the mRNA processing and splicing in the nucleus. Okay, you will study in the next year about the transcription. Transcription is the formation of RNA, mRNA from the DNA. Okay, so from the DNA, mRNA will be formed. That time, the first mRNA should be processed, then only become the functional mRNA. For processing, there are certain, before that time, there are certain RNA involved, that is called small nuclear RNA. There is another RNA called microRNA. MicroRNA regulates the gene expression. Okay, gene expression will be regulated by microRNA, and siRNA is there. siRNA is the small interfering RNA. So you will study in the next year biotechnology part RNA interference. Okay, in the RNA interference, this one helping for siRNA helping to silence the specific gene. Specific gene will be silenced by this type of RNA. So these RNAs play a crucial role in controlling the gene expression and maintaining the cellular homeostasis. Okay. Yeah. So last part, we will compare the functions of DNA and RNA. So DNA, it carries the genetic information. Okay. So all the genetic materials are stored in the DNA that consists of genes, various types of genes. Another thing is from the DNA, it can make unique copies. That is called a DNA replication. It creates identical copies of its own copy will be produced by for cell division. Okay, and also for the protein synthesis, the genetic code or the message will be given by the DNA. So these are the key functions of DNA. Coming to RNA, whatever the information taken from the DNA, that message will be in the form of mRNA. Okay, and mRNA carries the genetic code. That's what information transfer. Genetic code is taken by the messenger RNA. Then amino acid transport. We know the transfer RNA transfer the amino acid to the ribosomes for protein synthesis. Catalytic activity is also there. Who is doing the catalytic activity? rRNA is doing the catalytic activity for the formation of peptide bonds. And also gene regulation, gene expression or the protein synthesis will be controlled by RNA. Okay. So the last part, central dogma. Central dogma in molecular biology explains that from the DNA, what is produced? The RNA will be produced. From the RNA, what is produced? Protein synthesized. Okay. So this is the central dogma describes the flow of genetic information in the biological system. Okay, so here on the DNA only genetic information present. This genetic information going in the form of RNA and from the RNA to protein. So the protein will be produced. Okay, so the production of the protein is many, this flow will be there. It will not produce directly from the DNA. The messages will be given to the RNA or in the form of the RNA. RNA, then from the RNA protein synthesized. Okay, and also from the DNA, it can make its own copy, the DNA replication. So replication of the DNA is what? Synthesis of DNA. Okay. Copies of DNA will be produced. That is called a replication. From the DNA, RNA is produced. That is called a transcription. 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 And from the RNA, protein is synthesized. That is called a translation. This flow of genetic information in the biological system that is called a central dogma. Clear? Yes? Yeah. Now we are coming to reaching the last part of the session. So once again, we'll discuss compare the two nucleic acids, DNA, RNA. DNA is double-stranded, RNA is single-stranded. The sugar present, deoxyribose sugar in the case of DNA, RNA only the ribose sugar. Okay. Then the base pairs. Remember the base is present inside the DNA: adenine, thymine, guanine, cytosine. Here it is adenine, uracil, guanine, and cytosine. The DNA present only in the nucleus. There are certain exception case like in the case of some cell some cell organelles and also the bacteria and all it is there, but still this the new it is present in generally it is present in the nucleus, but RNA present both in the nucleus and cytoplasm. The functional difference: DNA, it is the information storage area, but RNA involved in protein synthesis. Okay. So that's all. Thank you. Thank you, all of you.